next item on our list here throughout 24 hours is some of the beautiful molecular landscape renderings by DigiZyme. Some of you might be familiar with that work. If you're not yet, you will be absolutely amazed by that multiple landscape and multiple scale work. We recently had Gail give us a presentation in our CNS talk series, and I will share a video recording of this because he couldn't be available with us today. So I'm going to screen share this particular video, and I hope you can see it. So Gail Medale, introduced by Andreas Bückler, which you saw earlier today. And here it goes. Enjoy.
So thanks, everybody, for coming in. I think it's time to start. Some people might still be wrapping up calls and they're going to join us momentarily. So everybody, welcome to this week's CNS Visual Showcase. I'm very happy to have a fantastic speaker lined up for today. We got Dr. Gail McGill with us. Gail is faculty and director of the molecular visualization at Harvard Medical School. And he's also the president and CEO of a company of DigiZyme Incorporated. Gail is going to talk to us today about knowledge synthesis through scientific visualization. And I promised you a visual stunner in my announcement, and I'm sure that's what we're going to get. So Gail is going to be with us for the full hour, and then afterwards we're going to have a meeting with Red Pill, Blue Pill, and Gail for the organ gallery. So without further ado, Gail, the stage is yours.
Yeah, well, thank you. Thanks for having me again. It's always exciting to get a chance to share one's work. As I was saying a few minutes ago, I wanted to also make sure to keep things fresh. So there are some for those of you who may have seen the presentation that I gave earlier, August 3rd or something like that, with a part of this group. I apologize. You may see some similar things. But what I wanted to do today is to focus on a bit of a broader interest that we think kind of almost as an umbrella over all the different work we do. And that's this idea of knowledge synthesis and mental models. So what are mental models? How do they relate to how we learn, how we think? And what is the unique relationship between visualization as an activity and how we create mental models of the science in our minds? And so that's kind of been, the theme that I've used at least to organize today's talk. So I'll start by sharing screen. And I guess I'll do the whole desktop. And can you see my screen okay? Yep. Are you able to see my screen? Yes. Yep. Okay. Wonderful.
Yes.
Thanks for confirming. Okay, so without going into, usually I take some time to introduce my team and the kind of work that we do. I've tried to really condense that so we could get, and I can bring as much of that back in the Q&A if there's interest. But in a nutshell, as Andy described, so part of my life I spend as kind of, as an academic. I'm a cell molecular cancer biologist by training, but I really haven't done kind of wet lab work for at least the last 10 years. I've shifted all of my research to more what is basically almost like cognitive psychology, learning sciences research. So we study how design decisions drive effective communication, educational visualization pieces. So quick examples of the kinds of things we're interested in. One of our grants, one of our networks, for example, is looking at how bringing teachers, learning scientists, and visualization practitioners together can lead to more effective creation of educational media and increase the overall credibility of those media. And again, these are just snippets of the kinds of questions we ask. Another thing that we do is we try to create kind of controlled visualizations where we try to tease apart variables in the visualizations that can affect learning. So this is an example of a study we did a number of years ago where we'll create 4 different versions of a very similar animation, changing some of the visual variables and using eye tracking on top of these to understand what students are looking at various stages. And This combined with things like adaptive assessment trees, where we try to see how learner misconceptions can also impact the way they process and learn from visualizations. So again, forgive me for jumping around a little bit, but these are just kind of a spattering of the kinds of questions we're interested in. And it's not just for, the molecular world, even though we've tried to develop, for example, a set of molecular visualization principles that should help to avoid misconceptions. One of the main ones from this chart that you can't see at the top, but we find that over 80% of students, this was testing over 1,000 undergraduate biology students, more than 80% of students think that, for example, molecules know where they're going, right? That they have intentionality in their movement, that a growth factor will go find its receptor, which is a problem, right? Because that is definitely a misconception that will ride through the curriculum and have significant impact on someone's mental model of how chemistry works. So again, whether it's molecular or whether we apply some of these same principles to things like studying how insects move and how to teach insect locomotion, this is the kind of thing that I do as part of my academic self and the kinds of papers that we publish there. So that's actually kind of the smallest part of my activity. The main part of my life is spent running the company that Andy mentioned, which is Digizyme, which I started over 20 years ago now. So the idea behind Digizyme was to not just have scientists collaborating with artists and designers to create effective visualizations, but actually to have the people who do this work be dually trained. So everyone at Digizyme is a scientist by training, graduate level, PhD level scientist, but also production trained in design animation. So it's the same people doing that work. One minute you can be reading a Nature paper, the next minute you're using, Hollywood type software to create an animation. And the reason I wanted to create that completely integrated team is that I felt that it really had a positive effect on the nature of the visuals that we created. And I will go into more of that later. So as part of Digizyme, again, we see our work being at the intersection of a number of different fields. Obviously, the science, an understanding of the subject matter, the technology aspect, right? Multimedia and understanding those technologies and the affordances of those technologies. What is the right type of media to develop to teach or communicate a particular concept? I'll come back to that later. Everything that comes from the field of design, of art and design. So what we know about how the visual system processes a display and leveraging all of those tools to our benefit. And then finally, what we know about cognition from the learning sciences, the cognitive psychology. Again, these are rich fields, but what we try to do is make sure that our work draws upon all of this information from different areas. And the kind of work we've done at Digizyme, again, I'm summarizing a rather large portfolio, but it's been not only for a number of different types of clients, biotech, pharma, academia, science museums, but also we've created a lot of our own internal products as well. And I'll show you some of our internal efforts as part of today's presentation as well. One of the things we've done to enable our work is to create our own software that, we don't just keep to ourselves. So Molecular Maya is available to the community and is used by a number of different teams. Some are competitors. And so the idea is to create a piece of software that leverages the power of the same software tools that are used for the Hollywood blockbusters. So very powerful, very give us incredible kind of simulation and aesthetic flexibility. But the problem with that software is that it was never meant for scientific data, especially for biological data sets. So we've built Molecular Maya to be able to quickly import data sets from various sources and to create coarse-grained simulations and visualizations that retain a high level of scientific accuracy, but also give us a fair bit of design flexibility. And again, all of this is by way just of background. So the last talk that I gave in early August was mostly focused on this idea of, what are some of the challenges in multi-scale visualization, right? So if you're depending on what to, and this was, of course, connected to some of the things that I know some of you are doing, like the hub map and other things. Visualization is not just a tool to create images, right? It is a process that forces you to collect, import, and kind of cohabitate multiple data sets from different sources in order to get, as I love this Max Planck quote, a comprehensive picture, literally, of what you're doing. And so that integration of data can either be from different sources, right? So when we're creating, let's say, a molecular or cellular animation, we might collect data from some of the areas that you see on screen. But it's also integrating things over different, dimensions. In other words, there's also the aspect of integrating things that happen over not just spatial, but also temporal scales. And I believe I showed this slide last time as an example of the complexity of molecular motion that happens at multiple time scales and at multiple spatial scales all at the same time. So that was kind of the theme of my last talk. Today, as I said, I want to focus on something a little bit, related, but different. And that is the idea of mental models, right? So what is a mental model? How do we think using mental models? How do we construct them? And what can visualization do? What is the kind of the special power that visualization and visual media have in seeding our mental models? And so as a little exercise for fun, sometimes I like to use this progression of slides. So I start with octopus, right? And so the idea is, by just giving you this word, I'm triggering in your mind, presumably, each and every one of you in a different way, what you think of when you think of an octopus, right? So if, it might look something like this. If you're a marine zoologist, again, you might have something like this. But, if you're a middle school student, you might think of this, right? Or maybe even this. So what if you're a nature photographer, right? You might immediately think of an octopus, but more in terms of how you're framing your subject. You might think about lighting. As a neuroanatomist, you might immediately have a vision of it through your lens, your mental lens that focuses on neuroanatomy or computational biology, et cetera, et cetera. And, sometimes it goes even beyond that, right? So you might even, as a Wall Street banker, be thinking of an octopus in this way, right? So the idea that I wanted to bring up with this little sequence is that even though for something like an octopus, where we can all literally get together, we could all get together in a room, in an aquarium, and look at an octopus together, we all bring our own mental models of what that means to us, right? And so in crafting a mental model, I would argue that, this is way simplified, but if we consider some of the steps that go into the formation of our mental models, probably the first step is sensory perception, right? So what do we actually see, hear, feel? Our window onto the outside world is probably one of the first steps. The next step is some of the cognitive filters, right? So this is not just raw information. We, of course, process that information in different ways. And that processing also meets existing information that's already in our minds, right? So for each one of these steps, you might consider some of the issues that you need to consider. For sensory perception, it's something as simple as, do we share the same senses? Well, not quite, not all the time, right? In terms of cognitive filters, again, we have differences, individual differences. And certainly in terms of our pre-existing knowledge, we all come to an experience with different backgrounds, with different experiences. So, the process of creating a mental model, even when we're all looking at the same thing, something we can look at, like a macroscopic object, can wind up being very different. So that's for the octopus, right? So now what if I say insulin, right? I would argue that now we're kind of shifting into a slightly different danger zone, right? So insulin no one's actually ever seen insulin, I would argue, right? I mean, you've seen models of insulin, right? You've seen maybe pictures of an insulin crystal that a crystallographer grew. You may have seen representations, but you've never seen insulin directly. So what if I say ribosome? Same thing, right? Or transcription initiation complex, or you probably know where I'm going with this, right? With increasing complexity all the way to, let's say, if I say a cell, every single one on this call has a different perceptual understanding of what a cell is. And it's probably, again, this complicated mix of all of your past experiences with what you perceive to be a model of a cell, right? So that's your mental model of a cell. And what I thought I would do in the next series of examples is show how we've used visualization to try and influence different audiences' mental models of certain topics. And I thought I would kind of start with literally K-12 and go all the way up to, scientists, skipping along the way. So one thing that I wanted to show is an experience that I had teaching sixth grade science for a few days, which was a lot of fun, a number of years ago. And, one of the things you read in the literature is at this level, unfortunately, biology is often taught, especially when it's cell biology, as you ask a sixth grader what a cell is, and they'll say, well, it's a container with a lot of complicated names in it, right? And that's basically the issue. And so I showed this to sixth graders, and I said, okay, these are some of the words you've recently learned. In a couple of years, you're going to have to learn some other big words. And then in a few years, it might look like this. At this point, the class is getting a little bit nervous, right? These are 12-year-olds. And then I showed them this image. And then I say, eventually it's going to look like this, which intentionally is completely freaking them out at this point. But the experiment is to then say that this is actually an extremely simplified version of the cell, and then to show them this image. And so the purpose of this little progression was to try to shift their mental model from these incredibly simplistic diagrams, which I think teachers, for the most part, are choosing to use because of their simplicity. And I'm not saying that they're not useful, of course, but the question becomes, what is the learning objective at that age? What are we trying to do with a 12-year-old student in explaining what a cell is? I can tell you what happened when I showed this image, which is that most of the hands went up in the classroom, which was exactly the response I was hoping for: engagement, excitement, and a ton of questions. And so I even went further. I told them, this is actually still a very simplified, dilute version of the cell. If you look at even just a tiny piece of it, it's much more dense, and it's constantly moving randomly. It's a total mess, right? Again, the idea with this progression was not to teach every single protein in this cellular landscape. It was not even to teach the basic organelles. It was to give them a feeling of the complexity of life at that scale that could trigger engagement and interest. So this is one example I wanted to show you. And by the way, when we take some of these principles to high school, this is an example of a very simple interactive learning module that we created. So a number of years ago, I co-authored with E.O. Wilson a digital textbook in partnership with Apple called Life on Earth. And we created over 500 of these interactive media learning modules. And this is an example of a learning modules. This is now high school students who need to know a little bit more about things like molecular families. As they click on the buttons on the left, we basically highlight not only the representative molecule itself, like a protein or a piece of DNA, but we try to give them context for not only the location and geography in the cell, but also the rough and relative abundance of some of these different molecular families. So again, the molecular families they have to learn, but typically the way they're shown in a typical textbook does not provide them with the context of where they live and how many of them there are. if we keep going higher in levels, and this I did mention, I think, in my talk before, but I thought I would quickly bring back this example because to me, it's a good example of how, mental models are still critical, even for experts, right? So I showed you kids, 12-year-olds, high school. I also, we do a lot of work at the undergraduate level. This is an example of a process that I'm about to show you that I would argue is complex enough that even experts working on it have a hard time creating a viable mental model of how it works. Okay, so that process is the way in which the SARS-CoV-2 spike protein drives membrane fusion. And this is a representative, by the way, of a larger class of proteins called class 2 fusion proteins, which work very similarly at the surface of HIV, flu, dengue, and other Ebola. In other words, there's a common theme in the way that many of these proteins work to drive membrane fusion. But I'll use SARS-CoV-2 spike as an example. So without going into the details of the process, the idea is that once the virus has attached its external receptor to the surface of a cell, and whether or not that fusion happens on the outside of the cell or if it needs to be endocytosed and happens inside, that's kind of detail we don't need to go in today. The key difficulty, the key puzzle, is the way in which this now tethered virus is going to be drawn closer and closer to the target cell such that the membranes will eventually fuse. Fusing two hydrophobic membranes is something that has a significant energetic barrier, right? It's not something that happens spontaneously. So the spike protein basically is the engine, the molecular machine that drives this membrane fusion. And it goes through a series of intermediates, again, that are known across other viruses. So on the left, you'll see there's what's called a pre-fusion intermediate, which is essentially what the spike looks like as it's binding to the cell. Then it sheds some of its surface cap, and it basically stabs the host membrane. So now you have a protein that is embedded in both the viral membrane and the cellular membrane. And of course, I'll show you a visualization of this in just a minute. And then if you look all the way to the right, after this protein has somehow refolded and fused the membranes, it has this intermediate known the post-fusion intermediate. So we have structural data, that everyone has structural data in the PDB, cryo-EM, other databases that represent some of these intermediates. But the process by which the protein refolds, that is the complicated puzzle. And so I wanted to show you the visualizations we've created over the last couple of years, looking at how this process might happen. The first step is that the spike unfolds and extends its hydrophobic peptide or platform into the target cell. So I'll replay this one just for a second. So notice that there's an alpha helical transition. This yellow helix extends itself, and in doing so, it projects part of the spike into the host membrane, which is the gray-blue membrane below. The next step is that this yellow helix will serve as a template onto which another set of helices will now refold. So the second alpha helical transition provides the motor force to drive the two membranes to merge together. And we've created a more detailed molecular model now in cross-section with an actual model of the membrane. So just to start at the beginning, right? So notice that ACE2 receptor is in green, and we have a couple of spikes that have just lost their protein caps. And now you see the initial extension. Now that the virus is tethered, it will now refold again. And in bringing together those membranes close together, it leads to what's called a hemifusion intermediate. The membranes are not totally fused, but they eventually resolve into a membrane pore, which will deliver the genome payload into the cytosol. That process that I just showed you is a coarse-grained simulation that we had to reconstruct, right? So it was like a big puzzle in space and time, where we had data from literally hundreds of papers, right, from different fields. So X-ray crystallography, cryo-EM, NMR, circular dichroism. I mean, all these different rich methods, but that only give you a little part of the story, right? And so in this case, I would argue that visualization is not just about communicating something that's already known, right, and making it pretty and engaging and clear. The act of visualization is actually the knowledge integration process itself. Yes, an animation is the result, but the process that we had to go through to bring all these data together into a common space was probably the most powerful aspect of the experience. And we were very fortunate to continue through collaboration to then do a cryo-EM tomography, such that, so initially we published this in Science now a year ago, and it was the basis for also explaining how these nasal inhibitors will block the fusion process. But we continued this work with our collaborators, and just recently this year published in Science Advances, where we were able to validate our modeled intermediates using cryo-EM tomography, which was a really exciting moment for us, because it showed that the work that we were doing with modeling and simulation was now validated with real tomography data. So again, mental models. I'm hoping, if you've never seen this visualization before, that your concept of how viruses drive membrane fusion may have been influenced or shifted based on seeing this visualization. The refolding of that protein is something that's rather dramatic, and that I would dare say, once you've seen it that way, it's kind of difficult to think of it in a different way. So visualizations are not only very powerful, but of course, if you don't get it right, they can also be quite powerful and dangerous in the other direction. My point today is simply that they form very fertile ground upon which we build our mental models and then understand and integrate further data. So one more example I wanted to show you, this time for a much broader audience, kind of a professional audience, but one across really many different fields. One of the things an internal project we've been working on over the past 4 or 5 years is, as the title implies, is how sustainable engineering solutions depend on the biodiversity of life. So this is part of a book that we're writing, and I wanted to show you a few examples of this, again, to try and influence your mental models of why do we need biodiversity, right? Other than the argument that it is almost a responsibility that we have, kind of the moral argument, we're trying to make a more specific argument about how much we are dependent on biodiversity, not just in terms of our health, of course, nature services and all kinds of different medicines that we derive from biodiversity, but specifically to meet the engineering solutions that we're going to need to develop in order to live sustainably on this planet. Many of those engineering challenges, it turns out, have already been solved by the plants, the animals, the microbes that live all around us. The only difference is they've already done the engineering field trials over hundreds of millions of years. So here are some examples of what I mean by that. There's a beetle in the Namibian desert. This is one of the most arid places on Earth, right? Almost no water. Well, this beetle every morning climbs up the dunes and sticks its butt up in the air, and then it waits. And so the question is, what is it waiting for? Right? Well, so we created this interactive that lets you explore the morphology of the beetle a little bit. And what you notice when you look at the elytra on the back is that it has hydrophilic bumps that are surrounded by superhydrophobic channels. And so what happens is that the Namibian beetle is basically a fog harvester. So one thing that does happen occasionally on the Namibian desert is you will get water-rich air coming in from the ocean. And this beetle, thanks to the chemistry of its back, is able to condense water droplets. By standing on its head, the water droplets trickle along its body all the way to its mouth and allow it to drink. And this has actually served as a model for human-based engineering of nets that also collect water from fog. So an example of a model of how we can look to nature. Another example that we use is dragonflies, right? So dragonflies are some of nature's oldest flyers. So they're kind of the black belts of the sky. They can fly forward, backwards. They can take off vertically. They fly up to 40 miles an hour. They can fly upside down. Amazing. Some of the oldest flyers. They've been around for over 300 million years. And they have a prey capture rate of 97%, right? So if you're a fly near a hungry dragonfly, bad news. You're probably not going to make it. But here's what I'm getting at. Most of us have seen dragonflies kind of from above, right? A picture of a dragonfly wing. And they have these beautiful kind of stained glass window patterns that you see. What you don't often realize is that a dragonfly wing is a corrugated structure. So if you look on the right, if you were to take cross sections along this wing, notice that the side cross section of this wing is not at all a smooth surface, unlike an airplane wing. Well, why does this matter? It turns out that dragonfly wings are shaped in this way because they lead to microvortices that form in the little valleys on the wing. And so without going into the details of it, these microvortices actually lead to a fluid-on-fluid design that improves the aerodynamics and reduces the amount of drag on the wing. And so this understanding, which we would not know this if we were not to study dragonfly wings, this understanding has led to the design of micro wind turbines that actually spin at much lower wind speeds and have advantages over the monsters that we're creating right now, right? So here, that's another example. Here's another example, soaring birds, right? What you probably have noticed over the last 10 years or so is that almost every airplane you've been on now, commercial airplane, has these little winglets at the edge of the wings, right? These winglets are a way to reduce drag based on the airflow that goes over the wings. And these have been basically inspired by soaring birds like eagles and vultures. So these mechanisms, I won't go into the engineering of an airfoil, but the difference in pressure leads to the air that is being pushed to the edge of the wing, leads to these vortices that create these dangerous wakes and that induce turbulence. And so these little winglets that were inspired by birds are now being added to almost every airplane that you can see. And then one more quick example, humpback whales, right? So what do we have to learn from these guys? Well, it turns out they have an incredibly efficient hunting technique called bubble net feeding. During this process, several whales are hunting together and emit a curtain of bubbles that essentially encircles fish. The fish do not swim through the bubbles, so they're kind of all brought together. And as these bubbles rise, the whales are doing incredibly sharp turns in the water. And they're doing it at angles, what's called the angle of attack, that would otherwise call stalling on an airplane, right? So stalling is when the angle of attack of the wing has gone beyond a certain degree that the amount of turbulence leads to the plane falling out of the sky. Well, whales have figured out a way somehow to avoid stalling at very high angles of attack. Wouldn't it be nice if we figured out how they did that? Well, if you study the aerodynamics or rather the fluid dynamics of the humpback whale flipper, you'll notice these edges, the four edges of the flipper have these tubercles along the front. Those tubercles lead to spinning vortices, very much like the dragonfly wing, and they reduce the amount of turbulence and push back boundary layer separation away from the pectoral flipper. So this has also led to the design of several new engineering devices, whether it's wind turbine blades or microfans in computers, right? But these are innovations that were directly taken from nature. And we were fortunate to be able to present this work at the United Nations General Assembly over the summer. So why have I shown you these examples? I intentionally wanted to do examples that were everywhere from, molecules to ants to cells to humpback whales. The subject matter specifically is less important than the way in which images shape our thinking. So I already suggested that there are at least multiple steps in the way that we create mental models. But then also the question becomes, as practitioners, how do we make decisions about the best and most effective way to communicate our science, right? How do we know what visual models to create to support our thinking? And there again, I think there are multiple steps to consider, right? So some of these steps involve deciding, of course, who is your audience and what are the key communication objectives. So our philosophy, at least at Digizyme, is that we can't create anything. We can't create an image, an animation, a VR app without knowing who it's for and how we intend to change that audience. Those are two critical things that you need to have a very good definition of before you start. Otherwise, you'll get lost in the design process, or you'll be seduced by other aspects like the aesthetics of the experience, right? So design decisions, in our experience, have to be driven by a clear definition of audience and communication objectives. Once you have those, there's still a question of, what do I build, right? So in the world of multimedia, you can create anything from a static image, an animation, an interactive, a game, an immersive virtual reality experience. That menu is open to you if you have the technical skills to pull it off. So the question becomes, for a particular learning objective, what is the best solution? And I'll share with you one of the resources that we're developing to help people with that. And then even once you've selected what that best format might be, you still have some tough design decisions ahead of you, right? Such as how to use all of the arsenal of design, color, contrast, motion in ways that support your learning objectives. So again, just, communication objectives have to do with audience setting and the best format, as I just mentioned, so we found that people who are not experienced in the design, multimedia or media design process sometimes have difficulty knowing what that menu is, right? Understanding all of the options available to them. And so what we're just in the process of creating is what we call a multimedia design atlas. And what it's meant to do is provide a series of categories of some of the different things that one can build. So it's not just a gallery of examples, it's a gallery of categories of media. So how is a still illustration in 2D different from a still illustration in 3D? Or how is that different from a linear animation or a slider interactive, et cetera, et cetera. And so for each one of these categories, we have a page that not only provides multiple examples of that design category, but also introduces some of the design considerations, some of the technologies, and some of the literature that actually explains the pros and cons of that. And so we're excited to be launching this probably a little bit later this year, and we're building this right at the moment. And then, of course, design decisions. So in closing, because I wanted to make sure to leave room for questions and discussion, I just wanted to kind of take a big step back and just remind people, in the same way that I was saying, octopus or insulin or cell, Magritte had it figured out decades ago, ceci n'est pas une pipe, right? This is not an actual peep. It's an image of a peep. And one could say the same thing of this, right? This is not a protein. You've never seen a protein. You've only seen someone's depiction of a protein. And so the question is, for those of us who are working, especially I would say perhaps in the life sciences, at scales of space and time that are outside the realm of our intuitive reach, right? Because we cannot inspect these things directly. These issues of design and mental models are absolutely critical because they form the basis of how we even think about our science. I would argue as far that they even shape the kinds of experiments that we imagine. And so that's why we're very interested in how visualization can help drive effective mental model creation. And I just thought I would throw this up. I mean, if you're interested in this sort of thing, I recently published some of these ideas in a worldview for Nature Microbiology. So yeah, thank you so much. And I'd love to take questions.